Okay, guys, thank you so much for coming. We're already on lecture 15, urinary system, and we're going to finish this and then just go immediately into midterm 3. So that's the good part. So here we go. Urinary system, guys. So this is the functions of the urinary system as a whole. Okay, and those are the basic things: the production of urine, right? It's got to produce urine, and that's done by the kidneys. There are only 4 or so organs in the urinary system: the kidneys, the ureters, the bladder, the urethra, and that's it. Okay, and the kidneys do the bulk of the work, and we're going to cover later on in a few slides just the functions of the kidneys, and we'll look at the structure of the kidneys because they're the main organ of the urinary system. So the ureters gather all the urine, and then goes into the bladder. So the ureters conduct urine to the bladder, and then the bladder stores it. The urinary bladder stores urine until it has reached the capacity. Then you get the micturition reflex to urinate, and then you empty. All right. And of course, the urethra transports to the outside. Okay. All right. Of the body. And so that's it. So those are basically the functions of the, but we'll focus a lot on this today. I saw in the module, you kind of had in the module, LS module, trouble with the anatomy and how you sequence the production of urine. Okay, moving from like the layers, like the cortex, and then you have those pyramids and the medulla, and then you have the sinuses, and we'll kind of try and go in detail on that later on. All right. So again, those are the organs of the urinary system. It's a really simple system, but it has very important functions. And we'll really look at the nephron, which is the unit of function in the kidney. All right, and then we'll look at how urine is produced. Okay, so continuing on with functions of the urinary system. Like I said, we're going to focus a lot more on the functions of the kidneys, because the kidneys are doing the bulk of the work. All right, so they eliminate waste, and this is metabolic waste. So the reactions that occur in our body, they are byproducts and waste that the urine gets rid of from the blood. Okay, so the kidneys filter about 60 times all your blood in one day, in 24 hours. So you can imagine somebody who's having kidney failure and they're waiting for a donor and they're having to go to dialysis. You see? So they get to go and spend quite some time doing that, getting the waste removed. Okay? So it's a very big function of the kidneys. So because the kidneys, filter blood and are able to see the concentration of the blood, okay? It is the organ that also concentrates, that controls blood volume, okay? Because the kidney, depending on how it retains or excretes ions, water follows it, okay? Water always follows the solids, right? Via osmosis, okay? So if your blood pressure goes low because your blood volume goes low, what happens? Your kidneys will retain more sodium. You will release a hormone called aldosterone, okay, that we saw in the endocrine system, all right? Aldosterone is one that will cause the kidney to retain more sodium. When you retain more sodium, you then retain more water. Water will follow the solids. And then you produce concentrated urine. Your blood volume goes up, blood pressure goes up, all right? So the opposite happens. So it's able to regulate blood volume that way, by either excreting ions and then water follows it and you have copious volumes of urine and blood volume goes down and blood pressure goes down. Or you retain more ions, water follows it, then blood volume goes up, blood pressure goes up. Okay? So if you get shot, if I give you a little scenario, if you get shot, then you start losing blood, okay? Then you will release aldosterone, the body will try to conserve more fluids because your blood pressure will start to drop when you're losing fluids, right? Blood. Okay, so it's related, the blood volume and the ion regulation. You know, the solids we're talking about are things like sodium, you know, potassium, calcium, all those ions that are in blood. Okay, you know, it will control the level of ions in there. And the same hormones, aldosterone and mineralocorticoids from the adrenal medulla also. Mineralocorticoids have to do, in fact, aldosterone is one of those mineralocorticoids. Right? So, extracellular fluid, pH, okay? You will notice when we talk about urine production and how the kidney it regulates the ions, there are sympaths and antipodes that will exchange base acid, you know, and base, you know, like the H plus. This will accumulate in blood from the respiratory system when you're transporting oxygen, right? So the kidney has the ability to lose H plus ions depending on the pH of the blood, so that the pH of the blood remains around neutral, 7.3, 7.4. Okay? Because once you've, you know, the cells have used the oxygen and they've given out the carbon dioxide and it's dissolved in blood, and you produce the carbonic acid, then the kidneys will regulate how much H plus, or the acidity of the blood. So that's how we do that. And I'll show you where that happens on the in the renal tubules. All right. So extracellular pH, you know, red blood cell synthesis. Remember, the kidneys produce erythropoietin. Remember this hormone? This causes the stem cells, the hemocytoblasts that will give rise to red blood cells to replicate in the bone marrow, erythropoietin. Okay? And we kind of saw this when we talked about the skeletal system. All right? What did we say? Whenever there's low calcium levels, okay, you release parathyroid hormone, and parathyroid hormone stimulates the production of active vitamin D in the kidneys. Active vitamin D is produced in the kidneys. And then what does active vitamin D do? What is the function of active vitamin D? Yes, it will absorb calcium in the small intestine. Okay? It helps to absorb calcium in the small intestine, right? And it's produced by the kidneys, all right? After PTH, okay? When calcium levels are low. So you could explain this if I ask about specifically about the functions of the kidneys, then you would talk about that. If I ask just the general functions of the urinary system, then you talk about what we said at the beginning when you say urine production and things like that, okay? And the functions of the 4 organs. Did you see what I'm saying?
No.
You don't? Okay, I could see that. So remember at the beginning I talked about the functions
General
of the
functions.
Yeah, that's of the whole system.
Okay.
Remember the system is going to
But
be
if you
written
ask for the functions of the
specifically of the kidneys,
Okay.
then you're talking about that, right? You see, I told you that the kidney is the main organ of the urinary system. The rest of the system really is for either storage or just conducting the fluid. And the kidney is doing all the work, really, right? The bladder just stores the urine, it doesn't do anything. You see what I'm saying? The ureters just conduct the urine to the bladder. And the urethra just takes it to the outside. You see? So the bulk of the, in fact, the meat and bones of the urinary system occurs in the kidneys. And we're going to talk a lot about that. And we're going to talk about hormonal control and how it is auto-regulated, which you kind of forms within the module as well. Auto-regulation is how the kidney ensures the glomerular filtration rate is constant. Because your blood pressure doesn't flatline, it always goes up and down. Okay? Good. All right, so the location of the kidneys, I mean, they are a retroperitoneal organ, which means they are behind the peritoneum in the abdominal, you know, and they are, you know, on the posterior aspect, okay, on the back. It's covered by a capsule that keeps it in place. So remember when we did the digestive system, after we kind of sort of pulled some of that peritoneum on the covering the abdominal cavity, we saw the kidneys right behind it. Okay, so it's called a retroperitoneal organ. course, it's behind the peritoneum, okay? Which is those, the parietal layer of the kidney. And it's, of course, surrounded by the renal capsule, which is a fibrous tissue that keeps it in place. And usually the right one is above the lower one. And that's just probably how the organs are arranged, okay, to fit in there. Okay, and then, of course, it's surrounded by perirenal fat. What might be the function of perirenal fat?
Protection.
To protect, of course. All right, and then the kidney has a hilum, you know. Okay, so the hilum is this area here where you got blood vessels entering, and you got whatever ureters, blood vessels going in and out, and nerves. And that part there where those enter is the hilum on the medial side. So that means that part faces inside. So this would be the kidney on the, so this would be the outside, and this would be the medial side, and then the other kidney would be in. So that's what it means, it faces inwards. All right? Okay, so that's the hilum there. And this is the kidney that we're talking about. So the hilum is somewhere here, like that. All right? And you can see the veins here, the inferior vena cava, renal veins, and then the aorta, abdominal aorta, supplying music, and then the perirenal fat over there. Okay, you should kind of look at these diagrams because these are actual cadavers, and this is the only chance you get to look at it, and you can see the perirenal fat and also the capsule, okay, the kidney. So this is it right there, the hilum. So this is where everything enters. The and exits, 4 exits, depending on what it is. Ureters and exit, carrying urine to the blood, right? So this is the part that you can have problems with. So the kidney really has got cortex on the outside, then the medulla has got the renal pyramids, okay, like that. And between the renal pyramids would be the columns, like that. So the columns are here, okay? And then they all project to that. And right here, at the renal pyramid, you have a minor calyx, okay? Because urine is kind of getting funneled towards the renal pelvis, right? Which is the exit of the ureter from the hilum, right? So you have the minor calyx, then you have the major calyx. I think this was in your question, what you call it, the module thing, asking you to, from the outside all the way going inside. It was in there, you guys had trouble with that. And then the major calyxes will project, towards the sinuses. Okay, the sinuses is sort of the innermost after the medulla. Then you have the sinuses after that. Okay, okay. So minor calyxes open into the major calyxes, which open into the renal pelvis, going towards the pelvis. The renal pelvis is the part of the ureter that exits from the hilum. Okay. So that's how you go from it. You see, you got the cortex on the outside, then you got the medulla, which is made up of the pyramids, and the pyramids are interdigitated with these columns, renal columns, in the middle, and I'll show you nice pictures where you can actually see that. And then the renal pyramids are kind of like funnels. They funnel into the minor calyx, and then the major calyx, and then the renal pelvis takes out to the ureters. Okay, so this kind of sort of look at that, minor first, major, then sinuses, and in the sinuses it's kind of like spaces. So the pelvis leads to the renal pelvis to the ureters. Okay.
So it's minor, major, renal pelvis.
Yeah, it's cortex on the outside.
Where's the sinus? Is it?
The sinus
Where's
is
the
just
renal
this.
pelvis?
The sinuses is kind of just the spaces, sort of just after the major calyxes, or somewhere around there, where you kind of get blood vessels and so forth. And how how the renal artery gets into the kidney, it will start to branch profusely. And these sinuses are kind of spaces where those arteries will branch out, and then of course the veins will branch together to form the renal vein. You see what I'm saying? So those sinuses is like a space. Okay, so and then that's where you have location of the branching of the blood vessels, that arteries getting into the kidney and you can imagine how much to branch to supply an artery to each nephron. Okay, so it's going to branch a lot. So it's going to come in through, and then the sinus, that's where it will start to branch. The renal artery will go into renal branches, renal artery branches. Okay, renal artery branches, then we go to the periphery. We'll talk about blood flow too.
Okay.
And when I show you a picture, it will come out nicely. Okay? Let me show you, like this one. You see what I'm saying? Cortex on the outside, then you got the renal pyramids, right? You see the columns are interdigitated between the renal pyramids, and the sinuses are here.
Oh, okay.
Sinuses are here. Okay? You see what I'm saying? So here, that's where the blood vessels come into here, and these are the sinuses. You see what I'm saying?
Oh, I see.
Those are sinuses. So one of the questions is, can you talk about the structures of the kidney starting superficial, going deep in the head, the cortex, the medulla, and the, you know what I'm saying, and the sinuses. So you have to start with the cortex is the most superficial, the medulla, and the sinuses. Okay? So these are the sinuses. They're not labeled here, but those are them. So there are sort of spaces where the branches occur and then feed to the, you see these branches I'm talking about? This is in the sinus. These are all the sinuses. Okay? Because this was kind of problematic in the module. So you see what I'm saying? So now this one is an even better diagram because it gives you all those things in sequence. You know what I'm saying? Okay, so you've got the major calyxes, then the minor calyxes, you're kind of sort of going to more superficial. And then you got the sinuses that I was talking about, remember, sinuses? So these are the minor calyx, and the papilla, which is right after the minor calyx, the papilla, because it kind of funnels into the papilla and then it goes into the renal pelvis, all right? After going to the major calyxes. So the renal pelvis is this bit here. Immediately outside, yes, it exits the liver. All right. So look at these pictures. These are actual kidneys, so that's what I like about those pictures. So you can look at those, and they are highlighted nicely. Okay, so the functional unit of the kidney, like I said, is the nephron. It's a series of tubules, okay, that start with the afferent arteriole going towards the glomerulus, which is like a tuft of capillaries. And then the efferent with an E goes out, okay? And then after you collect all that filtrate, then you have a system of tubules, and which will start with the proximal convoluted tubule, then the descending column, and then the ascending column, which make up the loop of Henle, and then the distal convoluted tubule, okay, which then goes to the collecting ducts, and then it goes to the eventually end up in the ureters in that fashion that we talked about. From the papilla, minor calyxes, major calyxes, you know, and pelvis. Okay? So I'll show you the picture of that, you know, so that's what it is. Afferent arteriole coming in from the branch of the renal artery. It will go into a tuft of capillaries like this, and that's called the glomerulus. The tuft of capillaries is called the glomerulus. And then the glomerulus is capped by what is called the Bowman's capsule, okay? Because after fluid exits the blood, the glomerulus is going to be in the cup, okay? And I'll show you a picture. So you get an afferent arteriole comes in, forms the glomerulus, and then the efferent arteriole is thinner. This is fatter, that's thinner.
Which one's the one on the left? Efferent?
This is the afferent.
Oh.
Afferent is towards something.
Okay.
Okay. And efferent is this one. Okay. So this is the Bowman's capsule. Okay. Like that. And then the Bowman's capsule will lead to the proximal convoluted tubule. Okay. So the Bowman's capsule and the glomerulus is called the renal corpuscle.
So afferent is before something.
No, afferent is before.
That's afferent.
Afferent with an A towards the glomerulus. So the afferent is going this way. The efferent with an E is going away from it. Okay? And the efferent arteriole is that one that leads to the peritubular capillaries. Okay? Because the peritubular, you know, when you filter blood, anything that fits goes. So you filter a whole bunch of the things you actually want to keep in blood. Okay? So what the blood system does is that the efferent arteriole will come back and surround all those tubules so that everything that was filtered that needs to stay in blood will come out, and that's what's called tubular reabsorption. And then we'll go back into the blood vessels. Okay? So the blood vessels that come from this efferent one, that come and wrap themselves around these renal tubules, the first set is the peritubular capillaries, and then the other one that is on the loop of Henle is called vesicerator. And I'll show you a picture so don't worry, okay? So, blood comes in through the afferent, goes into the glomerulus, and then fluid enters the Bowman's capsule, which is like a cup, and then everything is funneled into the proximal convoluted tubule. All right? Okay, so and I'll show you a picture of that. So, but on the whole, it goes into the nephron, fluid leaves the glomerulus, goes into the Bowman's capsule. The nephron then empties into the distal convoluted tubule, right? After you have the loop of Henle, and then this is the distal convoluted tubule. Okay, like that. Okay? And then this blood vessel will come and wrap itself around these. Okay? So the distal convoluted tubule then goes into the collecting duct, because there will be another collecting duct right here that collects all that. By the time it gets to the collecting duct, it's almost destined to be urine, unless if you release ADH, which is antidiuretic hormone, which will insert some water channels into the collecting duct, and then you retain more water. Okay. Other than that, mostly once the filtrate that ends up in the collecting duct, most of it is destined to be urine. Okay. All right. And then the collecting ducts empty to the papillary ducts. So there's a renal papilla, which goes into the minor calyxes. Remember, it said the papilla at that time, so those are the papillary ducts. The minor calyxes, the major calyxes, then the renal fluids, then the ureters. Okay, so good. I'll show you again the picture of the, the nephron, because I want you to know the parts that make up the nephron and how, the filtrate is conducted up until it's urine and then exits. Okay. Because really what you do with the kidney, what it does, it just filters everything from blood. Then it reabsorbs what it likes to keep. And then some things that may have escaped, through the filtration system that, and then went into blood, those things are secreted into the tubules, and then they become urine, okay? So you just filter everything, then you edit after that. So that's pretty much urine formation. After this, we'll talk about tubular reabsorption and tubular secretion. Okay, so it's filtration, tubular reabsorption, tubular secretion in the blood. That's all that there is to it. Okay, so again, you have trouble with these cortical, the kinds of nephrons they are. Okay, remember the kidney's got two major layers: the cortex on the outside and the medulla on the inside, right? So there are some nephrons that have short loops of Henle, short tubules. Okay, they're short. And they are located in the cortex mostly. Okay. It's just a little bit going into, those are called cortical nephrons. Okay. And these are the ones that just aid in the production of urine. Okay. Just sort of dilute urine. Okay. Because they don't have the ability to lose more fluid because they're short. Okay. Pretty much what's filtered kind of goes and becomes urine. Like a bit is reabsorbed. Okay. So 85%, so that's the majority. This was in the module, and you can have a problem with that, okay? So, and it gave you options, check all that's correct for cortical nephrons, then you would say it just aids in dilute urine, producing urine, and then it is, the most numerous, okay? 85%, which is almost all of it, okay? Only 15%, the only thing is that your module was saying juxtaglomerular instead of juxtamedullary, okay?
A lot of stuff was both different in the module versus the book.
Yeah. So I saw that a bit of confusion, so maybe, okay, I forgive you on that one. But it's supposed to be juxtamedullary. Why? Juxta just means next to. So it's the ones that are next to the medulla and they have the longest nephrons, okay? So the descending limb is very long, the ascending limb is very long. So there's more chance to lose fluid from those tubules, right, if they are long, because you get more chance of reabsorption. So they produce concentrated urine, okay, and that was in the module too. the medullary, juxtamedullary nephrons produce concentrated urine. Okay, and they're only 15%. Okay, because okay, I better get another. Okay, and so they have extensive thin segments, long, are involved in production of concentrated urine. Okay, so that was in the okay. This one is dilute urine because they are short. Okay, it's kind of like this. Okay, and then the collecting duct. So it's short, but the other ones are like long, like this, long. So more chance of losing fluid from it, right? So the juxtamedullary one produce more concentrated urine, okay? And they're less numerous. You see what I'm saying? Look how long this thing is. See what I'm saying? This is the medulla, right? So this is a juxtamedullary because you see how the renal corpuscle is right next to the medulla? It's next to. And all the nephron really is in the, because the kidney, what it creates this counter current exchange system, right? Okay? Which creates, this is more dilute, the fluid surrounding these parts is more dilute, starts about 300 milliosmoles, okay, up to like 1,200. There's some animals that really live in deserts. They can really concentrate their urine and really void almost nothing, so they retain more water. So sometimes they don't even need to drink because the kidney can retain all the water and using metabolic water.
So like camels.
Yeah. They actually, you know, how when you have reactions, some of these dehydration reactions and so forth that produce water, they have more of those reactions. And the kidney will retain all that water so that the animal doesn't need to drink water. It doesn't need to do that. Okay? So you see how it gets concentrated. So the longer the nephron, the more water is going to be absorbed. Because if it's concentration here, fluid, water is going to move from a less concentration to the high concentration, right? Via osmosis. So the longer the nephron loop, the more concentrated urine is going to produce, right? Because most of the fluid is going to come out. See what I'm saying? So that's what it is. And then you see these other ones are short, the cortical nephrons. And those are the majority of it, because humans produce dilute urine because we drink a lot of water anyway. Okay? So animals that don't have a lot of water, and they will have a bunch more juxtamedullary nephrons, you see what I'm saying? So that they produce very concentrated urine. So Bowman's capsule, you see, he's got the, remember it's like this. This will be the visceral layer, okay? Because it's very close to the glomerulus, and this will be the parietal layer. So that's what it's talking about. Parietal, visceral. Remember, the visceral layer is one that's called it's the visceral layer that has the podocytes. The podocytes are cells that have processes that have holes on them. Why? Because then you need to filter that so that the fluid, when it enters the Bowman's capsule, it will enter through those podocytes. And then it will enter into the proximal convoluted tubule, right? See what I'm saying? So there's podocytes. Okay, so the filtration membrane, which is just like the respiratory membrane, so the gases have to pass through that. The filtration membrane, the filtrate has to pass through that. Okay, so consists of the endothelium of the glomerular capillaries, right? Because the glomerular capillaries have little slits on them called fenestrae. Okay, so it has to pass through the endothelial layer of those glomerular capillaries. This is the tuft of capillaries like this, and this is Bowman's capsule. So the tuft of capillaries has the fenestrae, which are slits that open up and let the filtrate out. Then the visceral layer of Bowman's capsule, the one that is immediately surrounding the the glomerulus, that's the one that has the podocytes. And podocytes are cells that have processes like this that create slits, openings for the filtrate to go through. Okay? So the epithelia of blood vessels have a basement membrane, right? Okay, so you've got so it goes through the epithelia, then the basement membrane, okay, of the epithelial cells lining the blood vessels, the glomerulus. Then also it has to go through the podocytes. in the exam, if I ask you, can you list the 3 components, and then one will be for bonus, of the, you know, the filtration membrane, then you talk about that. You talk about the endothelium of the glomerulus, of the blood vessel, then the basement membrane, then the podocytes, which is the visceral layer of the Bowman's capsule. Then you are done. Okay?
What kind of question would that be?
I say the components of the filtration membrane. Okay? Components of the filtration membrane. Just like the respiratory membrane too, which we did when we did the respiratory system, right? Remember, just to go through the epithelia of the blood vessels, it has to go through the lining, the alveolar epithelium, the basement membranes, and so forth. Same thing here. So, just has to go through the endothelium of the blood vessel, which is fenestrate, little slits on it. Okay? These are slits. Okay? Openings. And then the basement membrane of the epithelia of the blood vessels, and then of course the podocytes, which is the cells that line the visceral layer of the Bowman's capsule. Okay, so those 3 things from the filtration membrane. Okay, so those are them. Okay, it shows you the podocyte with those slits. You see how the slits open up and then form those openings where the filtrate goes through. Okay, so pretty much anything that fits goes. So you're going to take me a lot of things that need to stay in blood. So you have to reabsorb that. Okay? There's a bunch of reabsorption that actually takes place. If you look at the composition of filtrate and the composition of plasma, it's almost the same, except the filtrate doesn't have plasma proteins because those are huge. They can enter, exit, okay, and then the blood cells. So that's what it is, guys. Okay. So remember those capillary beds I was talking about? I was saying that because you filter everything that is in plasma that you need, okay, you're going to have to reabsorb that. So that's where you create this peritubular capillaries and the vasulator that will come back and surround these tubules, so that when those things exit out of there, they are reabsorbed. That's why it's called reabsorption. Because they started in blood, now they go back to blood. They are being reabsorbed. So one thing I'm going to give you a warning on right now is that tubular doesn't mean renal tubules. It means blood vessels, okay? I didn't name them, so, they could have just called them, capillary, whatever, reabsorption. That makes more sense, right? But they said tubular, okay? So tubular reabsorption is not referring to the distal in the renal tubules, pretty much. It's referring to blood vessels, okay? And if it's tubular secretion, it's saying that the blood vessels are secreting it back into because it's something that is waste, but kind of escaped the filtration process and ended up in blood, so it's got to go back to the tubules. Okay.
When it's talking about the peritubular capillaries, isn't it talking about this in proximal?
What's that? The peritubular capillaries are those capillaries that come from the efferent arteriole and come back.
But they are talking about the distal and proximal tubules, convoluted tubules.
Okay, the peritubular capillaries are capillaries surrounding the distal convoluted tubule, proximal convoluted tubule. Yeah. And the proximal convoluted tubule and the distal convoluted tubule are part of the nephron ductwork. Yeah, and so forth. So the reabsorption that's occurring here is not going into the tubules. It's called tubular reabsorption, but it's not going to the tubules, it's going to the blood vessels. That's what I want you to get. Okay?
But
you see what I'm saying?
It happens at the tubules?
Well, yeah, because if I was to draw it for you again, so this is the afferent arteriole. It comes in, then it forms the glomerulus, and then the efferent arteriole. Okay, and then we have our little Bowman's capsule here. Okay. Okay. So this is the proximal convoluted tubule. Proximal. I'm going to abbreviate it. Loop of Henle, descending, ascending. Okay, and then distal convoluted tubule.
Right.
Okay. So what happens is that the efferent arteriole, okay, is containing blood, right? It's going to come back and surround all these tubules. Okay.
And the capillary bed.
Yeah. This is the peritubular capillaries. Okay, and then there are special ones that go to loop of Henle called vasa recta. Okay, that go like this, and then this is going towards the venous side now. This is still the capillary side. These are the vasa recta. Okay, the peritubular capillaries are up here, surrounding the distal and the all those ducts and the proximal and so forth. Okay, so those are the peritubular capillaries. So you start off with the glomerulus, which is the capillaries. Then the efferent arteriole leads to these peritubular capillaries, which are these ones up here. And these are specialized peritubular capillaries called vasa recta, and it was in your module 2. That one they asked you about it, and they had problems with that. They asked about the special tubular that go on they called it the nephron loop, they called it something funny like that. They were talking about loop of Henle, okay, and you had to fill it in the answers, okay? But that's what it is. So each glomerulus is fed by an afferent is going towards the glomerulus. So this is the afferent right here. R with an A. Okay, and this is, do you notice I did it on purpose? This is fatter than this. What would be the purpose of that?
So you get more blood to it.
What's that?
So you get more blood to filter.
Yeah, how is that? Because it does what? What is the effect? Is it increase?
Filtered.
Yeah, it's being filtered, so how? That's what I'm saying.
How it just is.
Well, no. Because when you've got large volume coming in, okay, because this is wider, so it can carry large volume of blood coming in, and then you crimp the hose so that only small come out, then pressure goes high, so it filters. That's the only way. See, that's the reason. So I didn't draw it by mistake. This is fatter than that. And auto-regulation involves altering the diameter of this, the afferent arteriole, because that creates the pressure, the filtration pressure. Okay? And blood pressure is fluctuating. So, and glomerular filtration rate is got to be constant now. It can't be bothered with the up and down of blood pressure. Okay? So, you either increase that or decrease it depending on the blood pressure, so that the glomerular filtration rate is constant and you filter your 60 times of blood. In a day, you go 60 times, your blood goes through your kidneys. 24 hours. Okay, so that's auto-regulation. You have problems with that, it was in the module as well. But I want you to note the differential between the afferent and the efferent arteriole. It creates pressure that allows the filtration to happen. Okay? Very good. All right, so your blood pressure in the glomerulus is high because the arterioles are high resistance, of course, vessels, especially that the efferent arteriole is much smaller. So the smaller the diameter, the higher the resistance. It's just like on a hose, you know what you do on a hose? If you want to have a spray, you put a spigot at the end that is smaller hose. If you just have the hose pipe, the whole of the hose pipe, it will just trickle like that. It just comes out, it doesn't do anything. But if you want it to have more pressure, okay, you don't have to open the tap more. All you have to do is put a thing that is smaller hose. Yeah. Or cover it, and that's what we used to actually back home. We didn't have those funds. I used to go like spray it, and then somebody would crimp the hose while you're watering the garden, and then you let's go.
Good times.
We used to do that all the time, you know. There you go. So afferent arterioles are larger, so that's why I wanted to show that I didn't do a mistake than the efferent, so that you can create that pressure. That differential creates the pressure, all right? And then the fluids are forced out. Everything's forced out, except the big, cumbersome, plasma proteins and red blood cells and so on can't fit, so they don't go. They stay. Okay? So in the exam, if I ask about how the pressure in the glomerulus is created, you can talk about that, right? Yep, that would be like a bonus question. So this is it right here. It's an abbreviated, nephron showing you the podocytes and the filtration. And you can see the fenestra on the glomerulus. And you see the distal convoluted tubule comes back and in very close proximity to the afferent arteriole. Okay. And it creates what's called the juxtaglomerular apparatus, which we'll talk about in a bit. Okay. The juxtaglomerular apparatus actually detects flow. So you can do auto-regulation, blood flow. And then it also produces a hormone called renin, which is involved in controlling blood pressure. Okay, and we'll talk about that. So there are special cells called macular denser cells on the distal convoluted tubule and granular cells. Sometimes they're called juxtaglomerular cells on the afferent tubule. Okay, we'll talk about that in a little bit. All right, so arteries and veins of the kidney. Arteries and veins of the kidney. So, like I was talking about, and I'll show you a nice diagram that shows blood flow through the kidneys. We're coming in through the renal artery, and then it will branch in those sinuses we're talking about earlier on. And then it, then branches into the afferent arteriole. So it will branch many times until it gets to the afferent arteriole, all right? Because each one has to supply each nephron, and there's a bunch of nephrons in there. Okay, so that's a very well organized system of blood vessels. It has to be, because how many nephrons are in the kidneys? A lot. Each one has got its own afferent arteriole. Come on now, right? So. And then they supply the glomeruli, okay, which becomes the glomerulus, the tuft of capillaries surrounded by Bowman's capsule. So the afferent arteriole then supply this one I was talking about. The peritubular capillaries surrounding all those distal proximal convoluted tubules, and then the vasa recta. Why does the blood vessels do that? Because you have filtered a whole bunch of things that need to stay in blood. So all those things that have been filtered into the renal tubules have got to be reabsorbed into blood. And this is how you do it, through these, okay? Through this vasa recta and peritubular capillaries, okay? Which come from the afferent.
So peritubular capillaries and vasa recta have to do with
Reabsorption, yeah, from the tubules, okay? That's why I was just giving you a forewarning that when we start to talk about tubular reabsorption,
there's a difference. Between just absorption and tubular reabsorption?
I know there's not a difference. I mean, tubular reabsorption, the first instinct is to think, okay, it must be the tubules, renal tubules they're talking about. But it's not. It's the blood vessels. Those are the blood vessels. So when they say tubular reabsorption, they're talking about reabsorption by these guys and these guys. Okay? They could have said capillary reabsorption, but they didn't, okay? I didn't name them. So I'm just telling you right now, so you know that. Okay, because it can get confusing when you say yes, tubular reabsorption and we're talking about renal tubules and you think we're going into the tubules. No, we're not going into the renal tubules, we're going into the blood vessels.
Reabsorbed back into the bloodstream.
Back into the bloodstream, not into the renal tubules. But I didn't name them, okay? So they did it. So, and then the vasa recta and so forth will lead to the venous side, and then it will form the venules, and then the venules will join to form branches of the vein, and then the branches of the vein will become the renal vein, and then it goes back into the vena cava, so inferior vena cava, right? Going back to the blood, to the heart, okay? So that was also a problematic question in the blood flow in the kidneys, starting with the renal artery and going back to the renal vein, okay? So this is what I was talking about earlier on. The distal convoluted tubule comes back into close proximity with the afferent arteriole, and it forms the juxtaglomerular apparatus, made up of specialized cells on the distal convoluted tubule called macula densa. Okay, they're sort of enlarged cells. And those cells, they're very much in close proximity to the granular cells on the afferent arteriole. They are kind of specialized cells of the epithelia of the afferent artery. Okay, the one that's going into the glomerulus. Okay, sometimes they're called juxtaglomerular cells. Okay, so these because they're next to the glomerulus. Yes.
So the vasa recta, it seems to go into the peritubular capillaries. Run into each other?
Oh, they do, yeah. Because the peritubular capillaries give rise to the vasa recta.
So it's just a matter of location?
It's just a matter of location. The vasa recta are specialized because whereas the peritubular capillaries are kind of convoluted like this, surrounding all those distal proximal convoluted tubule whatnot. Now, the vasa recta are kind of straight. They form a loop like this.
Okay.
and then around, and then loop of Henle comes down like that. Okay, and the vessel rector go like that. Join, 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 join. Okay, and this guy here is the capillary side, and this is the venous side. This goes into the renal venules, and then the branches of the renal vein, and then those branches will come together to form the renal vein and out of the kidney. Okay, so really the vertebral system and the vessel rector are continuous. It's just the same. They just specialize because of location. Yeah, differences in where they are located. Okay, so let us know, of course, the juxtaglomerular apparatus, what it is made up of, and the kind of cells that it's made up of. Okay, the distal convoluted tubule with the macular denser cells, and the afferent arteriole with the granular cells, sometimes called juxtaglomerular cells. Okay. Why? Because they're next to the glomerulus. Okay. You can call them granular cells. It's okay. I don't mind. Okay. Granular cells. Okay. So know that juxtaglomerular apparatus, and the function of it is to produce granule. Okay. So if I go back, it becomes black and white. Oh crap.
I think it's like that when you pull them up off of.
Sometimes it goes black and white, sometimes it's color, so I don't know. I can't put heads and tails on it. Okay guys, so here's what I was talking about. This is the blood flow, and this was, sometimes I ask this question in the exam, you know, and it was in your module too. So you've got the renal artery coming in at the hilum of the kidney, right? And then it's going to branch. It's going to form branches that are formed in the sinuses, right? Remember those sinuses, those spaces. Then after it forms those renal branches, then they'll divide until they form the afferent arterioles. Okay. The afferent arterioles will go to the glomerulus. Okay. Then the glomerulus goes to the efferent arteriole. Efferent with an E. The efferent goes to the peritubular first, capillaries, then the vasa recta. You see how it's continuous? Peritubular here, peritubular, and then vasa recta down here. Okay? The vasa recta is on the venous side, you see what I'm saying? And then it's on the venous side, and then it forms a venule, and then, you know, before you know it, it joins the renal branch, the renal vein branch. The renal vein branches join to form the renal vein. Okay? And then the renal vein goes to where? It's number 10. Wait, I told you just a second ago.
Renal vein.
Where does the renal vein go to? Yeah, inferior vena cava. Okay? Inferior vena cava. Okay? Sometimes I have questions that ask me to track things like in the digestive system. You could be tracking a nutrient that doesn't get digested.
In the test?
Yeah, in the test, in this third test. In the third test. Okay. Sometimes I do that. Or you could track urine, you know, production in there, or blood flow in the kidneys. Okay, so that's how it goes. Okay. So now urine production. Now urine production really is those three processes. Okay, we're going to talk about that, and then I'll just show you how the renal tubule absorbs, and then also, you know, I mean the renal blood vessels absorb and the transport on the renal tubules and the kind of transporters you have, and then we'll talk about hormonal control in later. Okay, but pretty much the urine production is the filtration. Okay, then the tubular reabsorption, and then the tubular secretion. Okay, all right. Like I was saying, 60 times a day, your blood is filtered. Your 5 to 6 liters goes through your kidneys 60 times in 24 hours. Okay, so filtrate contains, it's almost similar to plasma, you know, except some of the plasma proteins. Okay, so contains plasma components except those big plasma proteins. They can exit those little fenestrae. Okay, so it will lose water, it will lose nutrients. That's why when you're diabetic, your urine is sweet, because it's got glucose in it. Why? Because there's so much of it in blood. Not all of it is going to be reabsorbed and it just gets voided.
Isn't that how they tested for diabetes? Is they
Yes.
actually
Back in the
doctors
day, they
actually
used to drink
tasted
it.
urine?
If you don't have gonorrhea and stuff like that, you're all good, man. In fact, urine is sterile. It only gets dirty when it comes out. It's sterile, yeah. So it's clean. Otherwise, you'd be infected, unless if you have a urinary tract infection. Then that won't be good. You can't drink that. Okay? It's not for human consumption. But they just tasted it, and on the tongue, it's sweet because of the glucose. And then you get your diabetic, and then, you know. Of course, the doctor will go through some symptoms first to really, before he tests the urine, because he will know that the diabetic guy will present with the symptoms. You say, oh, you might have that. So the urine contains the metabolic waste that we're talking about, okay? But most of the filtrate, of course, because the filtration is not discriminatory, it's just the size of the molecules. That's all that discriminates them. Otherwise, all the goodies come out. You get to reabsorb all those. In fact, most of it is reabsorbed. And then you produce only like 1% is going to be urine. See what I'm saying? And then, so this is what it is. Filtration, tubular reabsorption, and tubular secretion. And this tubular means blood vessels, okay? That's what I was trying to say earlier on, okay? It does not mean renal tubules, no. All this reabsorption is occurring into the blood vessels, the peritubular capillaries and vasa recta, okay? And then all the secretion is coming out of the vasa recta and all those peritubular capillaries going back into the renal tubules. Because the filtration process doesn't care, you know, just sometimes things that need to be in the urine escape the filtration and they end up in the blood vessels. Those things have to be secreted out. Okay. So when we talk tubular here, we're talking blood vessels, okay? Forget the renal tubules. Okay? So that's the only thing that I want you to talk about this. Boom, boom, boom. See, there you go. You get your blood coming through the afferent, then you get your filtration. Okay? And then boom, boom, boom. You gotta reabsorb a bunch of that stuff, right? And you reabsorb it back into the blood, and then a few things that may have escaped, and then they go back into the tubules. And then anything that ends up in the tubules, urine. Okay? So you can see here plasma and urine, the differences. You know, so pretty much the filtrate is very close to plasma. I wish I had a table comparing filtrate and plasma. You see that there's not a big difference. It's almost like plasma except for a few things, okay? But now urine is really different because now we're just getting rid of the waste and excess ions and stuff like that, okay? And you can kind of look at this later to see how it goes. So urine formation, of course, like we said, filtration based on the pressure differential, then filtration, tubular reabsorption. You know, reabsorb back into the blood what you need, and then anything that's been secreted. You see how you have the two arrows here? You gotta reabsorb a lot of the stuff, and then you secrete just a few of the things that is kept, okay, and back into the tubular system, okay, and that becomes urine. So pretty much just describe these three. So this is all there is to urine production, okay. So again, we kind of sort of repeat the whole thing. You see what I'm saying? Most of the filtrate, 99% of it is reabsorbed. It's going to go back into the blood vessels. Because the filtration process doesn't discriminate. Anything that fits goes through. Right? So, glomerular filtration rate is going to be kept constant, okay? It's the rate at which filtrate is produced per minute. Okay? That's where you get into auto-regulation. The filtrate is, you know, pretty much like plasma, really, okay? Except for a few things, like the blood cells, the platelets, and the blood plasma proteins, they're humongous. They can't enter through those, they can't exit through those fenestrations and so forth. So, most of it is reabsorbed. And then you only produce 1% urine. So, reabsorption is a very important process, right? Because you've got to take back into blood all those things. In fact, when you beginning to see kidney failure, you know what they do? They start to see more nutrients and all sorts of stuff in your urine. You see what I'm saying? When you're starting to get renal failure, because you're not reabsorbing all those things. Okay? So sometimes, you go to the hospital and you're prone to just, you start to lose color. After, because you're not removing all the metabolic waste properly, it will start to discolor you. Okay? Yeah, so forth. So filtration process, of course, responsible for urine formation. And then we just reabsorb. So what really happens is that, and I'll show you about how the pressures that actually result in filtration. Okay. First of all, we talked about how we have the fat or the big diameter afferent and the small diameter efferent. Okay. So that creates the pressure. Okay. In the glomerulus, and then fluid exits. Okay. When you put fluid in a container, such as the Bowman's capsule, you start to build hydrostatic pressure, right? Okay. And also, as you remove fluids from the blood, it's going to become concentrated. So you're going to have osmotic pool that's going to oppose the filtration. See what I'm saying? And the hydrostatic pressure, called the capillary hydrostatic pressure, inside the glomerular capsule. Because the glomerular capsule is filling up with fluid, so it's going to have pressure too. Okay? So the pressure in the glomerulus minus the colloid osmotic pressure, pulling back the fluid, because once the blood becomes concentrated, it will now have osmotic pressure, right? Remember osmotic pressure like we did? And once the glomerular capsule is filling up, it will also have capsular hydrostatic pressure, okay? And that will oppose filtration. So the final result of filtration is the glomerular pressure minus the osmotic pressure in the blood vessels, and also minus the capillary hydrostatic pressure. And then the difference is about 10, and I'll show you. Okay? So that's what this is talking about. Okay? Glomerular capillary pressure minus the osmotic pressure in the glomerulus, which is the pool, once the blood is concentrated, because it has just lost a bunch of fluid. And because you're putting fluid in the capsule, in the Bowman's capsule, it's going to have hydrostatic pressure that's going to start to push out. You see what I'm saying? Whenever you fill anything with fluid, there's pressure there. See what I'm saying? So the difference between the capillary, glomerular capillary pressure and the other two pressures is what is the filtration pressure with the final pressure. Okay? So this is what it is. So the glomerular capillary pressure, which is pressure in here, okay, minus the blood colloid osmotic pressure. Because when you remove fluid from the blood vessels, it's going to become more concentrated. Okay? So any concentrated fluid is going to start to pull, osmotic pressure is going to start to pull the fluid back. Okay? So that's another osmotic, and it can go to 30. Okay? It goes to 30. And here we've accounted for capsular hydrostatic pressure. Because as fluid fills up in here, okay, I'll put that to show what. As fluid fills up in here, it's going to start to create opposing pressure. You can see that, right? It's going to start to have capsular hydrostatic pressure. So you have to subtract that. So that's about 10. And so 50 is the pressure you're building up in your capillaries. And then you subtract the pressure in the capsule itself. Okay? And then you subtract the pressure of the osmotic pool of the concentrated blood now. Okay? And the end result will be 10 milliliters of mercury. That's what the filtration pressure is. Okay? If I ask you to explain the end result filtration pressure, you'll be able to do it, right? By saying that it will be the difference between the glomerular capillary pressure, okay? That's created by the difference between the afferent and the efferent arteriole. And then it also subtract the blood colloid osmotic pressure, because when blood is concentrated, it has a tendency to pull fluid water towards it. Okay? And then the capsule is also filling up with fluid, so it's going to have capsular hydrostatic pressure. So that's going to oppose, okay? So the final pressure is 10, okay? And it's enough to filter. All right, so now urine production. This is the auto-regulation part that you're following. Because the blood pressure doesn't flatline, and so the kidney cannot be having all that fluctuating pressure coming into and out of the glomerulus. It needs to maintain the same filtration pressure, okay? So autoregulation is responsible for that. It's regulating itself. Okay? And it happens by altering the diameter of the afferent arteriole. Okay, because that's what creates the pressure differential in the first place anyway, right? So, under normal conditions, it will maintain almost constant glomerular filtration rate. So, the amount of fluid rate you're producing per unit time, per minute. Okay? So, there's two components to autoregulation, right? The myogenic part refers to muscles contracting, smooth muscle, okay? Of the blood vessels, right? That's myogenic. And then remember the juxtaglomerular apparatus I talked about earlier on? Because the flow is coming through the afferent arteriole. And it is the afferent arteriole and the distal convoluted tubule that form the juxtaglomerular apparatus. Okay, so that will have a sense of the flow of fluid into the glomerulus. So that's the tubulo-glomerular feedback. Okay? Because that's where it's coming through anyway, the afferent arteriole. Okay? So it's also edited by the flow that, the juxtaglomerular apparatus are specialized cells that will detect how much blood is flowing into the glomerulus. Okay? So if the blood pressure is high, you're going to get a lot of blood trying to come through, right? Because your blood pressure is high. So therefore, then the juxtaglomerular apparatus will sense the flow has gone up, and then it will try to reduce it by decreasing the diameter of the afferent arteriole. You see what I'm saying? And if the blood pressure is weak, maybe you've lost some blood, you've been short or whatever, then it will increase the diameter of it so that more blood goes through to maintain filtration rate constant. I know it's a lot of stuff that I'm bombarding you with. So sympathetic stimulation, of course, decreases renal blood flow. Because what does it do? It causes contraction of smooth muscle. Okay? If you have a smaller afferent arteriole, you're going to have small blood going in there. A small amount of blood. Okay? So just know the two components of autoregulation, the myogenic, which is the smooth muscle contracting, and also the flow rate measured by the juxtaglomerular apparatus. Okay? Okay, so this is the tubular reabsorption. All I want you to understand is that, when you're reabsorbing stuff, okay, the tubule is going to be like that, okay? So these are the cells, okay? So, and this is the filtrate. This is the filtrate. We just filtered it. So this is the tubule, the proximal convoluted tubule. So what is going to happen, this membrane on the inside is what's called the luminal membrane or the apical membrane, okay? So this is luminal. Because it's in the lumen. It's in the lumen is opening, right? Luminal or apical. And then this is the basolateral. Why? Because the basement membrane is there, and then it's the lateral. It's on the lateral side. So basolateral membrane. So anything that comes out of the renal tubules has got to cross the luminal membrane, has got to cross the basolateral membrane. That's what this is talking about. Okay? Do you see what I'm saying? The cells lining the renal tubules. Okay? And there's more cells on this side too. Okay. This is the nucleus. See that? So don't worry about all those music cosmos and whatnot, okay? Just know that the transported substances that move through all these membranes, okay? The luminal membrane, and then the basolateral membrane, and then it just enter the endothelium of the blood vessel, right? That's it, guys. That's how things are reabsorbed, okay? And then calcium, magnesium, and them, they can go between the cells. You see, between the cells here, see this, between the cells, I mean, there's a bit of distance between the cells. So the ions, because these have to be reabsorbed, most of them, they don't even need to go through those membranes with the transport systems. They just go through, they just go through between the cells like that. That's what's called paracellular, okay? Or, the paracellular pathways. That's what it means, those ions. And I'll show you a picture of that whole thing, okay? Okay, so tubular reabsorption, most of it is reabsorbed, okay? And I'm going to kind of go through quickly because we talked a lot about this, and I'm going to show you the different transport mechanisms that go through the luminal membrane or apical membrane and the basolateral membrane, okay? And those are sympotors. You know sympotors, right? We did that in AMP1. You know antipotors, you know that go in the opposite direction. And all that, facilitated diffusion, all those transport mechanisms that we've done before. Nothing new here, okay? I'm going to show you how you move all those nutrients, glucose in them, okay? So that's why, because tubular reabsorption is the major process, you see what I'm saying? Because why? You're absorbing 99% of that filtrate, okay? Let's forgive him because he's done his thing, you know what I'm saying? So, this is what I'm talking about. This legend is wrong, okay? This legend is wrong. So, whenever you see, so this is the sodium potassium pump creates it's just wrong colored. The green is sometimes antiport and sometimes symport, and the yellow is sometimes antiport, sometimes symport, okay? So that's the only thing. So don't worry about that. Worry about what's really happening. So this would be a symport, you see what I'm saying? Because it's getting sodium and the amino acids, okay? So this is the lumen, right? You see what I'm saying? So to start off the whole process, this is interstitial fluid, okay? So you have the number one, the sodium potassium pump will generate the the sodium gradient, okay, where it will pump sodium out and pump potassium in, right? It pumps 3 sodiums out, 2 potassium in, like it always does, right? So you're creating a gradient here, so that sodium you want to move from here into the cells. You see what I'm saying? Okay, so that's the number one. You create a sodium, you put the sodium into the interstitial, into this interstitial space, and then it's absorbed back into blood vessels. Okay, and whatever sodium is in here is going to want to come in here. Do you see what I'm saying? That's how you osmotically pull the sodium out of the filtrate. Do you see how that's happening? Okay, guys, I gotta move on. Okay, because we have created that sodium, and there's a bunch of sodium in here. Okay, the second step is that sodium is going to want to come out of the filtrate, because this is the lumen, right? This is the luminal membrane, and this is the basolateral membrane. You see, this is the cells lining the tubules, the renal tubule. Do you see that? Okay, so now the sodium is coming in here by simple diffusion, because there's no less sodium in here. Okay, it pulls with it amino acids and glucose and so forth. Okay, that's the second step. So this is the symport. Do you see that? You symport sodium and you are going in the same direction. Amino acids and glucose, they are all symported with sodium. Okay? And then, number 3, because, again, another sodium, this is an antiport now. Okay? Number 3, as sodium is drawn into the cell because it's coming out here from the lumen, it's a positive charge, right? A positive charge, whenever sodium goes into the cell, what happens? Depolarization. Because it's charged, right? So in order to prevent that, because you want to get rid of the pH, remember we said the function of the kidney is to regulate pH, okay? So the urine, that's why the urine is acidic, okay? So you take off the H plus, which carries the same positive charge, and you take it out. So there's that antiport, which will create electrical neutrality. You see what I'm saying? Makes sense? Okay? So this is an antiport, because it's sodium in, H plus out. And then there's electrical neutrality. We are all neutral. We're not depolarizing the membrane. Okay, because otherwise it would depolarize. Okay, because we don't want to do that. Okay, by the way, all those glucose and amino acids on number 2 that come in here, they are through facilitated diffusion will come out. Okay? With this facilitated diffusion. And then the glucose and the amino acids and nutrients will be absorbed back into the peritubular blood vessels. Okay? Okay, number 4, because acid is base, like carbonic acid, it's got H plus and HCO3. which is the base. The negatively charged part of it is the base. And the base, again with an antiport, the base comes out, because that's why the urine is acidic, right? Because you have already taken out the H plus on 3, and here you're bringing out the base on the, you know what I'm saying? This is the negatively charged one, okay? And then, because the base is negatively charged, you've got to antiport it with chloride, which is negatively charged, so that you have electrical neutrality. Otherwise, you would depolarize or hyperpolarize the membrane, okay? So this is an antiport again. Okay, water just goes by diffusion. It just goes through, okay? You see how water just goes through? It just goes through. And it just follows the ions, okay? All right? And then number 5, water just goes through. Because as long as you remove the ions, water follows the ions. And then water is reabsorbed too, okay? So those are the processes. And then you remember the paracellular pathways I was talking about? The calcium, magnesium, potassium, and all those ions, they don't even need to be transported here on the transporters. They just go through the paracellular pathway, okay? That's the space between the cells. It doesn't even need transport. Because you got to reabsorb the whole bunch, and urea some also a little bit, and you know, goes between the cells. Okay? So that's the whole 6 processes. If I wanted you to talk about these processes and so forth.
I have to work.
Oh, sorry. I'm sorry, we kind of going over a little bit. But this system is a little complicated. I won't go through this. It's just renal secretion. Anything that escapes the filtration process doesn't come out and remains in the blood vessels. It's got to come out. And that's the tubular secretion, okay? So that's the minor process compared to this. So maintaining medullary concentration gradient, okay? Pretty much the vasodilata are responsible for that counter-current exchange. Because this is what I was talking about. The kidney builds osmolarity from the outside, going deep into the cortex, and it starts from about 300 milliosmoles. And in there, it will be in the deep layers of the kidney, the medulla, it's 1,200 milliosmoles. So that means water will come out of the renal tubules, right? Because water will come from high concentration to low concentration, right? Because this is got more solute than water. So it will just move from the renal tubules. So the longer the nephron, the more fluid can be absorbed out of it, because it's deeper because of the osmolarity. So the vasodilata are the ones that use that counter-current exchange, because the vasodilata dip down into the loops of Henle, okay? And they absorb more fluid because it gets more concentrated, and fluid comes out of the tubules, okay? So that's the counter-current exchange. And the urea, in order to create that, concentration in here, it moves out of the collecting duct and moves back in here, okay? Just so that to keep that concentration high, so that water exits, okay? And then antidiuretic hormone, of course, antidiuretic hormone only affects water absorption from the tubules. And what it does, it sets water channels called aquaporins. And then it doesn't affect the ions at all. Okay? So that's what I want you to know. And like we did in the endocrine system, antidiuretic hormone is actually produced by the hypothalamus, right? And then it goes through those axons into the vesicles of or the terminal vesicles that terminate in the pituitary. And then the pituitary just releases it. Remember when we did the neurohypophysis and so forth back in ANP1? That's what it is. And it was in your module 2, and you had problems with that. Okay? The DH is produced by the hypothalamus, but it's secreted by the pituitary, posterior pituitary. Okay, so that's that. And it inserts these aquaporins, and I'll show you how it does that in a little bit. Let me show you how it does. I'm sorry, I kind of have to sort of finish this up a little bit. So ADH, if the blood osmolarity goes up, which means it's got more concentration, okay, then you absorb more water, and then high concentration in blood means there's less water to dilute blood contents. So that's high osmolarity. So when blood volume goes down, it becomes high osmolarity because there's no water in it, and so the solutes are concentrated. So you release ADH. ADH causes reabsorption of more water to come back into blood by inserting water channels called aquaporins on the collecting duct and the distal tubules. Because they're normally impermeable to water, okay? And the opposite happens when there is high blood volume, so it's low osmolarity because it's more dilute because of the water. So you don't release antidiuretic hormone in that case. So you have diuresis, and then you lose more water. Okay, remember the G proteins that from your endocrine system. Okay, this is a protein-soluble hormone, water-soluble hormone, so it attaches, it excites G proteins. And then the alpha and gamma subunits will split from the alpha subunit attached to GTP, and then it will cause production of cyclic AMP. Cyclic AMP causes these vesicles. See these vesicles containing the water channels? They are placed onto the membranes, and then water comes out. Okay? Because normally you see the membrane doesn't have water channels on it. That's the collecting duct and the distal tubules. They're not permeable to water normally, but when you release ADH, it will insert these water channels in there. Okay, so that's that. So that's how the thing works. And the renin-angiotensin system, aldosterone system, controls the blood pressure. I'm gonna kind of go quickly here. So the renin is produced by the juxtaglomerular apparatus, and it converts. There's a hormone called angiotensinogen, which is produced by the liver, and then angiotensinogen is converted by renin into angiotensin 1. And then there's angiotensin converting enzyme, ACE. That's why ACE inhibitors, blood pressure medication. Okay? The angiotensin converting enzyme converts angiotensin 1 to angiotensin 2. And then angiotensin 2 causes the secretion of aldosterone, okay, from the adrenal medulla, which then does what? It causes more absorption of ions of sodium, and then you retain more water. Okay. All right. So that is released when the blood volume goes down, or and you need to retain more fluid, so you retain more ions. Okay. So I will pass through that renin-angiotensin system. And then atrial natriuretic peptide is what is released when the blood, when there's a lot of blood and the atria gets stretched by too much blood coming back to the heart, it will release atrial natriuretic peptide, okay? And then it will go to the kidneys and will cause the kidneys to excrete more sodium, and then you lose more water, okay? Natriuretic means uresis is lost through urine. And Na, that's why Na is the beginning of the word for sodium in some other language, in Latin or whatever. Okay, so that's why the symbol for sodium is Na. So natriuretic, you lose more sodium in the urine, and then you lose water. Okay, as long as the ADH is maintaining the osmolarity. Okay, because of the water content. Okay, so this is atrial natriuretic peptide. And then this just shows you the female and male bladder, okay? And then as usual, compare the systems. So I'm sorry it took a little long, but the kidneys, I got a lot of stuff, and I didn't have a chance to split it into urinary system 1 and 2. So, okay? Anyway, we'll revise. When we do the revision, I'll go through the things I want you to retain, okay? All right, thank you, guys.